Good evening, everyone. Am I audible? I hope you all are doing well. Welcome back to day 2 of our 1 week hands on workshop on network pharmacology, molecular docking, pharmacokinetics, and molecular simulation. On behalf of Thyme Phaeto Biomed Private Limited and AdGrow Foundation, I warmly welcome all our speakers and participants. Thank you all for joining us again today. I hope you enjoyed yesterday's session and learned something new. Today we will start learning about molecular docking. First, we will understand the basic theory of molecular docking. After that, we will with all the software that will use during the practical sessions in the coming days. So I request everyone to keep your laptop ready and carefully follow the instructions given by our experts. If you face any problem during the software installation, don't worry, our team will help you. Without taking much of your time, I would now like to invite our respected Dr. Ghanshyam Parmar to begin today's session on molecular docking. Sir, over to you.
Yeah, thank you so much. So, without further delay, I'll starting the session with the understanding of a theoretical aspect of a molecular docking. Okay, what actually you are using the various software and you done docking, but what actually running behind the software. Okay, software done its job. Okay, but what actually behind the software, everything is happened. Let's understand by just simplest way. Okay, I'll this session is divided in a two part. So initially I'll explain the theoretical part of a molecular docking. Then in next session, Mr. Jay Churasama will explain you or help you out to installation of all the software which I mentioned yesterday. Okay, that software you required for the molecular docking that will be installed in your system. Okay, so I will not take much of time. I just briefly explain you everything about the molecular docking, then we'll continue for the software installation. So let me share my screen first. I hope my screen is visible to all.
Yes, sir. Please start.
Yeah. So basically, let understand what is a molecular docking. Okay. I give you a brief some common example that based on that you can easily understand what is a molecular docking. In the simplest way, I can say that if we have a one lock, okay, and you have n numbers of keys, okay, you are finding which key is most suited for this lock. Okay, so this is the simplest example. By this example, you can easily understand that the which ligand molecule best fit with your selected protein. Okay, so basically we are predicting the preferred orientation of ligand when it bound to the target. Okay, we are just predicting how our ligand it fit into the active site or cavity of the protein. So that is a fundamental of a molecular docking. It is a technique that predicts the preferred binding of a ligand to the active site of a target protein. So in first of we have to understand what is the component of a molecular docking. The two main component which require mainly is a receptor and ligand. So in yesterday's session, I explained everything about what is receptor, what is ligand. Okay. So usually receptor or a protein or a target is a 3D structure which is we can obtain from the PDB database. Not only from the PDB database, one of the most common database used are called as a UniProt also. Okay, from UniProt, you can also find the uh, target or a protein. Okay, along with that, on UniProt, you will find all the detail about that particular protein or target or receptor or enzyme, whatever you are in a search of. You will find every detail regarding uh, this your target. Let me just divert you to the particular site. Then you can easily explain. You can easily understand how this uh, databases look like and how this database work. I'm just sharing my entire screen so that you can easily understand that, or you can easily visualize all my screens. So first, that is a PDB database, which is RCSB, which is a protein data bank. From this site, you can find all the proteins of your interest. Say, for example, by in the search area, you can find whatever the terms you can enter, whatever the term you are searching. Okay, or either you have a defined protein ID you can directly add to that ID and you can get the database or you can get the protein. So I am writing directly a code. So I'm interested to study the protein target 6LU7, which is actually a crystal structure of COVID-19 protease, which is complex with the inhibitors. Okay. So in this website, you will find the PDB structure with its features, like whether it is a X-ray crystallographic structure, what is the resolution. Okay. So it is experimentally derived structure. Okay. Which is you are going to use in your study. So you can find all the detail about the structure. You have to go through the various parameters that I will explain in a next subsequent slides. Like you have to look into this Ramachandran outlier, side chain outliers, that you have to check for the specific characteristic of a protein. Based on the parameter, you can select the protein, like resolution, which method is used to crystallize for the crystallization or prediction of the structure, whether it is X-ray diffraction, NMR, cryo-EM, which method is used. Based on that, you can select the structure, from which organism it has been derived. Okay. Based on that, you can select the structure. Here in this section, you will get the idea about the where its structure has been from, it has been derived, or related publications detail is available here. Okay. So you can explore this website and you can find many a thing which is most interesting to you or suitable to you. Another website that is called as a UniProt. This is also one of the database where you can find your interested protein, okay, or interested target. Okay. So I am, say for example, I am selecting the insulin. It will ask you the which form you want to display the result, either in table or column form. So here in the left hand side, you will find a detail about this protein, whether you are interested in from the human organism, a mouse, rat, bovine, zebrafish, according to your interest of your study. You will get all the detail about the protein. Okay, just I'm clicking on the human and I'm selecting, say for example, this one. Then after selecting, you will find all the detail of your interested target or protein, like its taxonomy, subcellular location, disease variant, expression, interaction, structure, family domain, everything, sequence, everything is given in this database. So before selecting of your target, you must have to understand what is your need, what is your target behavior, what is your target characteristic, which particular cavity or which particular active site you are looking for. Everything you should have to first thoroughly study. In this section, by clicking on the structure, you will find a structure of the protein, your interested protein. And from here, you can select the PDB ID. You can filter out by the method, whether you are looking for the EM structure, means cryo EM structure, modeling based, NMR based, predicted, X-ray based, whatever your interest. Okay. Or either you are looking for the experimental structure or predicted structure. If you are looking for the experimental structure, you have to select the PDB. Or if the experimental structure is not available, then you have to choose the AlphaFold, Swiss model, this kind of database where the predicted structure or homology modeling based structure are available. You can select like this. But mostly in our routine practice, we select the crystalline structure, which is mainly derived by the X-ray crystallography, and we have to look for the resolution of the structure. Okay, and you have to select the structure according to your requirement. Similarly, for the ligand, either you are choosing your own ligand which have you designed or synthesize in your lab. Okay, draw the structure by using various software like CamDraw, CamSketch, anything. Or either you can derive a ready-made structure from the various databases like Campbell, Drug Bank, PubChem. By using that database, you can directly download your structure or ligand. The next step, which is a docking process. Once you have receptor and ligand, now you have to move toward the docking process. So what are the general steps for the docking process? You have to first prepare the protein, which is called a protein preparation step. And then you have to also prepare the ligand also. So what is this? I just briefly explain you, because in a subsequent experimental or hands-on training, Mr. Jay will explain you everything by step. But I'm just explaining you briefly. In protein preparation, you have to download the protein structure from the in PDB form from the PDB database. You have to check the orientation of the structure. Okay, you have to either add up hydrogen, you have to add the charge, everything, and you have to optimize the structure of protein as well as the ligand. Once your protein and ligand structure are optimized by adding charges, by adding hydrogen, deleting the water molecules, okay, then you have to move toward the next operation, which is called as a sampling. Means explore the different possible orientation and conformation of the ligand in the binding site. Okay, you have to perform the docking and check whether your docking is run completely. After running completion of docking, you will find that most probably a 10 or 20 conformational orientation of your docked ligand into the protein. You have to check which one is a best dock or which having the best pose. Okay, along with that, you have to compare the docking score and RMSD value.
हेलो
Once the yes ma'am.
जी वो आंटी आई थी साथ वाली हमारी नेबर है तो मैं उनके पास बैठी हूँ वो बात कर
Anything?
रही थी तो
Hello?
मैंने किया था
Hello. Okay.
Please continue, sir.
Okay. Sorry. So after the scoring, you have to evaluate each pose using the scoring function and to estimate the binding affinity. Once the scoring you have done, you have to rank the selected molecules or rank the pose based on the score and select the best binding mode. How you select this? I'll explain you each and everything by subsequent slide. So, but what actually docking predict? Docking के करने से क्या हमें मिलता है? It gives you a binding pose, orientation and conformation of a structure into the binding cavity. It gives us a binding affinity or a scoring of your ligand. Either you are doing virtual screening or a single compound screening. It gives you a score of a binding in form of a binding affinity. It gives you information about the how ligand is binding with your protein in form of an interaction. Okay, whether it is hydrogen bond interaction, hydrophobic interaction, ionic interaction, pi-pi stacking. Also, it gives about idea about the important residue involved in your binding, which are you going to check after your pose has been revealed, or compare with your standard drug, how the drug is interact with the which residue. So you have to compare with your ligand residue, docked residue, and whether it is, you have to check whether the your ligand is actually fit into the desired space, or it bind to the other allosteric site. That can be confirmed by the docking. Okay, so there are three types of docking which we generally do in our research work. First is called as a rigid docking. Okay, mostly most of the people are doing the rigid docking, in which the both receptor and ligand both are rigid, both are stationary. Okay, while in another type, a flexible docking, where the ligand is flexible. Ligand have ability to orient in such a way that it can fit into the receptor, but here receptor is rigid. Okay, receptor is not flexible, receptor is rigid. The third and most important technique or important type of docking, which is called as induced fit docking, which is accepted and also a time consuming docking technique, which is called as induced fit docking, where both receptor as well as the ligand are flexible. So it gives us a best conformational pose or a dock pose, and with the highest accurate score. So this is considered as a one of the good technique for the molecular docking, but it is time consuming. The next aspect you have to keep in mind, the scoring function. So it is a mathematical functions that estimate the strength of interaction between ligand and protein. So different software have a different scoring function. Suppose if you are using software like Autodoc Vina, then mainly work on a force field base. Sorry, Autodoc. If you are using Autodoc, then it mainly work on a force field based scoring, which is a physics based or physics function based scoring system. While if you are Autodoc Vina, which is an empirical type of scoring system, which mainly work on the basis of experimental knowledge or experimental work done. Based on that knowledge, the scoring function has been given. So mostly Autodoc Vina, then Gold, that are work on the base of a empirical system. While consensus scoring, which used a multiple functions, either combination of force field and empirical. In empirical, there are two types of way. One is knowledge based and one is a statistical based. Okay, so many of the different software use the different scoring function according to their algorithm. Okay, so as I told you, that common interaction involved like hydrogen bond, hydrophobic, ionic, pi-pi stacking, and van der Waals are the common interaction observed in the docking process. The key point which you should have to keep in mind that docking prediction or docking predict how and how well our ligand bind to the target. It does not give 100% accurate result, but useful insights. And always validate docking result with the experimental. हाईएस्ट स्कोर आ गया, हम ऐसा समझ लेते हैं, we think like that कि highest scoring is the best ligand, but it's not like that. We have to validate it with the, again, by doing redocking and then go for the experimental validation. Okay. Let's move to the next slide. Let's understand how the protein structure and ligand have a different features. Okay, I already explained you everything. Like protein structure is you can obtain from the PDB database. So it is a large molecule made up of amino acids. That 3D structure determine its function. And in drug discovery, protein, usually enzyme or receptor, are considered as a target. I'm not going in much detail because you all are aware about the what protein structure, like primary structure, secondary structure, tertiary structure, quaternary structure, and its features. So I'm not going in detail. But here, one thing you have to keep in mind that the important resources from where you can get the PDB structure or your target structure or target protein, like PDB database, protein data bank, which mainly contain the experimental structure, which are derived by the X-ray crystallography, NMR, or cryo-EM methods. Also, you can search the various other sources like AlphaFold, Swiss model, where you can get the structure which is predicted structure, which is mainly when used when you don't have a experimental structure is available. Then you can go for homology modeling, and you can design your protein or design your structure by using the Swiss model. Okay. Then ligand, already we discussed in our previous session. Ligand is a small molecule that bind to the protein, and once it is bind, it change its or it modulate its structure, and it gives a protein function. So ligand may be either from natural source, either synthetic, or either maybe a design one. Okay. So indigenous ligand called as a natural molecules, which naturally present and you can take as a in your experiment. So that are natural molecules. Synthetic, which you synthesize in your lab, okay, which are man-made molecule. Then co-crystallized ligand, which ligand bound in the protein structure already available in the your PDB structure, which is called as a co-crystallized structure, which gives you about the reliable pose and idea about how it interact with the protein. And fragmented ligand, where the small chemicals fragmented use as a starting point in a drug design. So generally fragmented based drug discovery, where the fragment is used as a ligand. Further, the fragment is modified by addition of a various functional group, or I can say the R group, into the fragment and design a new molecules for the or designing of a new drug entity. Regarding protein site, okay, the as we also called as a active site or binding site, binding pocket. These are the synonym terms which is used for the protein binding site. Okay, this area, or I can say the groove, which is actually present in a structure of the protein or a space available in the protein where our ligand fit. Okay, so the features of this structure or cavity are important for understanding, which decide how your ligand fit better. Okay, so before going to before performing the molecular docking, you should have to understand the which binding site or which binding pocket is suited for you, because as you know, the protein having the quaternary structure, okay, and they have a different orientation and arrangement. So due to this orientation and arrangement, or bonding between the helix and seeds, beta seeds, they form a certain cavities. So these cavities are of a different size, different characteristic, different feature, different behavior. Okay, and based on this, their features, you have to select which is the best suited site for you, binding site for you. For that, you have to go to the literature review, and you have to also explore this database like PDB database, Uniprot. Okay. Then based on these interactions, you can find which binding site or which binding pocket is favorable for you. So this binding pocket or a binding site is specific region on the protein where ligand bind, is often a pocket or a groove which comprise of amino acid contributing to key interactions. So based on these amino acids, which is available in this pocket, decide your interaction. If the binding site having the higher non-polar amino acids like leucine, isoleucine, valine, phenylalanine, tryptophan, methionine, then there may be a possible of a hydrophobic interaction. If this amino acid having the polar or uncharged like serine, threonine, glycine, then tyrosine, then there may be a probability of a hydrogen bonding between the ligand and this amino acid. Likewise, positively charged, negatively charged, aromatic amino acids. Based on this, there may be a possibility of ionic interaction, pi-pi stacking. Okay, this kind of interaction can be possible. Beside this, there is also possible of a pi-cation interaction, which is between the aromatic ring and cation group. Pi-pi stacking, that is possible between the aromatic amino acid and your ligand contain aromatic ring. Then van der Waals, which is actually a weak short range attractions or short range interaction. So this kind of understanding gives you idea about how your ligand fit into the binding pocket. I just briefly explain you all these interactions. Already I explained you everything, but by this slide you can easily understand how the interaction can be or key force that stabilize your binding. So as I told you, the interaction type are electrostatic, polar, hydrophobic, aromatic, hydrogen bonding, van der Waals, halogen binding, and metal coordinates also. So I'm not going in detail with this. I'm just explaining you briefly that in hydrogen bonding, where the hydrogen of a donor hydrogen is form a bonding with the acceptor. Say for example, a hydrogen bond between a hydrogen donor and electronegative atom like nitrogen, oxygen, and it is a very strong bonding, and it decide whether your ligand have a specificity in a particular binding site, and it is directional and distance dependent. Some hydrogen bonding may be a long distance, or some hydrogen bonding may be a short distance also. So the length of this hydrogen bonding decide how the ligand is strongly bind to the site. the distance between the atom or distance of a bond, if less than 2 angstrom, is considered as a strong binding. More than 2 angstrom are considered as a loose binding. Then hydrophobic interaction occur between non-polar group, generally in a hydrophobic pocket. Leucine, valine, isoleucine, phenylalanine are imparting these hydrophobic interactions, which is mainly driven by the release of water molecule from the binding side. Then ionic interaction, which is occur between oppositely charged, positive and negatively charged, like carboxylic group and amide group. So it is mainly considered as a long range and strong interaction, which is important for binding and orientation. Pi-pi stacking, I already explained that it occurs between the two aromatic rings. Then pi-cation interaction, halogen bonding, van der Waals, and metal coordinates. These are the type of interaction which is possible in molecular docking. Let's understand how the binding affinity and scoring functions are work in a molecular docking. How we can interpret the binding affinity and scoring function at a different space, so that it gives you a easy idea or easy understanding how your ligand is binding strongly to the binding site or binding cavity. So keep in mind that lower the binding or more negative the binding score, having the strongest binding affinity. So the binding affinity is measured in a form of a calorie per mole in form of a binding energy, delta G, which is called as a Gibbs free energy of binding. So based on the thermodynamic process, delta G is equal to delta H minus T delta S, where delta H is enthalpy change due to favorable interactions, and entropy change due to desolvation and flexibility in an absolute temperature at Kelvin. So more the negative delta G, having the higher affinity. As Hemant sir yesterday explained that the delta G is reported as a score, kilocalorie per mole, from 0 to minus 10. Different software have a different scoring function. In some majority of software, you will get the scoring from 0 to minus 10 or more than minus 10 also. In some software where the scoring is done differently, so you may get a higher scoring function also, or different range also. So according to the software, you have to decide which scoring function you are using, and based on the rank, you have to decide the binding affinity. So scoring function which estimate the binding affinity of your pose or conformations, they approximate the delta G using mathematical terms used to rank pose and prioritize the compound. Once the compound is docked, you will get the docking score, which will decide whether you get the favorable or unfavorable interactions, and your scoring function will rank whether the compound is best or whether it is worst. Lowest the score, considered as the best compound. So type of scoring function, mainly force field based, like physics based energy terms, mainly Autodoc or Autodoc Vina, are give the scoring function based on the force field, while gold score, cam score, this kind of software use the empirical, which is called as a knowledge based scoring function, which is derived from the experimental data. Other software uses different kinds of scoring function like knowledge base, which is mainly another type of function similar to knowledge base, but here it is statistical base. Then consensus, which is a combination of a multiple scoring function. So main factor which affect the binding score and binding affinity is quality of a protein. How you select a quality protein. So as I told you that based on the database and based on the certain criterias, like how it has been derived, whether it is X-ray crystallography, NMR, what is the resolution, what are the Ramachandran outlier. So based on these characteristic, you have to select the quality protein. So if you have selected good quality protein, then you will get the best score. How the correct protonation state has been applied. During protein preparation or during ligand preparation, correct protonation state should be necessary. So that can be achieved by using, for ligand, you can use Avogadro, or you can use also a Autodoc same, which optimize the ligand. Also for a protein, you can use Autodoc or BioVia Drug Discovery Studio. You can use that kind of software for the correct protonation. Means you have to add the charge, you have to remove the non-polar hydrogen, you have to add the polar hydrogen. Okay, this kind of state is, or optimization is necessary for the good binding affinity and for the score. Then ligand conformation and tautomerism. You have to check whether the ligand having a right conformation. You have to check whether the binding site and its flexibility. You have to check the solvent effect, and scoring function limitation is also necessary. All these factors are affecting your binding affinity and scoring. There are certain limitations, like scoring functions are approximate. It is not that higher the binding score having the good biological activity. Keep in mind that, okay, it is not always true that if the score is higher or a best score, say for more negative, so it is not necessary that the ligand is the best. You have to check for other properties also. May not account for all entropic effect. Direct function may give a different rank. Need experimental validation, and better for ranking than absolute prediction. Common softwares which are mainly or common scoring function according to the software wise are mentioned here. I already explained it. I'm not going in detail. So keep in mind that good score suggests a good pose, but biological activity depend on many other factors. So keep in mind that. Just I'm briefly explain you the how the molecular docking workflow is possible and how you can perform the molecular docking. So first step is a protein preparation or target preparation. Then second step is a ligand preparation. Third step is define your binding site. For that, you have to define the coordinates. You can define the coordinates or a size of the box, grid box, which mostly or entirely cover your binding pocket. Right now, you are might not be able to understand, but during hands-on, you will easily understand what I am talking about. So first step is target identification or protein preparation. in which we have to remove the water molecule, add hydrogen, assign the bond order, then add hydrogen and minimize the energy of a protein. Then we have to go for ligand preparation, then grid box generation, or defining a binding area. You can choose, if you don't have an idea where to dock, you can choose a PDB structure where already co-crystal ligand is already docked. Find the coordinate using BioVia Drug Discovery Studio and use that coordinate for the right binding space or binding area. Next step is you have to perform the docking step, where you have to choose your appropriate software and your scoring function. Set the algorithm like exhaustiveness, whether you are working on a genetic or molecular basis. Set the parameters, exhaustiveness, number of mode, energy range. That in many of the software, like say for example Schrodinger, BioAir drug discovery, where these all the parameters are by default. While you are using Autodoc or Autodoc Vina, these all parameter you have to set by yourself. So this is the main difference between these some softwares that you might have to come across during using the different software. So what is exhaustiveness? Means exhaustiveness is a particularly higher the exhaustiveness, then there is a higher conformational changes of a ligand, possible conformation in your binding pocket. The highest the exhaustiveness, it gives you an accurate binding pose. Then you have saved your prepared file. Then you have to go for the dock simulation, where you have to run the calculation or docking calculation. Based on this, you will get the or you will find a different orientation and conformation of ligand in the binding site, and also you will get the score according to the your binding pose or along with the RMSD value. So in this section, you will find a scoring function and RMSD value. So you have to analyze the score as well as the RMSD value. As Hemant sir said that the score -7 up to the -7 is considered as a druggable molecule, and the scoring higher the scoring means more negative the score with the good candidate as a drug candidate or druggable substance. Also, we have to check the RMSD value for the best dock pose. Those conformation having the RMSD value lowest are considered as a best docking pose. Then visualize your docking pose by using a various software like PyMol, BioVia Drug Discovery, LeakPro, and you have to visualize your best pose. Suppose you are get the 10 pose out of your docking, you have to choose first pose according to this result and visualize in a different visualization software like PyMol, BioVia Drug Discovery, Chimera X. These kind of softwares are used for the visualization purpose, where you can visualize best strength pose, inspect interactions like whether it is hydrogen bond interaction, hydrophobic, pi-pi stacking, is there any steric hindrance are there. You have to check all these features, then confirm the pose is biologically responsible. Then next, after the getting the best pose and right confirmation, you have to go for validation. Okay. For that, you have to either redock your molecule again to validate whether you are getting the same RMSD value, Then dock non-ligand and compare with it. मान लो कि आप if you are using a protein which having already a co-crystal structure or co-crystal ligand binded already or docked along with your protein, then you have to extract out that ligand, redock it, and compare with your desired ligand, and check whether RMSD value is less than 2.2 angstrom. If RMSD is less than 2, then it confirms that your interested ligand bind a exact where the you desired. Along with that, you have to check the edimate property, drug likeliness analysis, then molecular dynamic and simulation for stability purpose, and binding free energy calculation. That ultimately all this gives you a best ligand or a druggable ligand or hits for your study. So in interpretation of docking result, I already explained everything. So I am just skipping this slide. The last slide, which gives you an application of molecular docking. So from computational insight to the real world impact, the docking can be used for the drug discovery and lead optimization, for the understanding the biological mechanism, how the enzymes and receptor can work, whether it is the inhibition, whether it is the activation, how the signaling pathway can be work, or generate a hypothesis, and for experimental validation. Also, it can be used for the structure-based design or a structure-based drug design. We can design a novel compound which is tailored to the binding site. By understanding of a binding site, we can design our own molecules based on the interaction, whether it's a hydrophobic interaction, hydrogen interaction, pi stacking, and we can design which amino acids are involved. According to that, we can design our molecules. Also, we can improve the potency, selectivity, and pharmacokinetic properties also. By changing the functional group, we can improve the potency. Also, we can improve the selectivity. Suppose we are changing the functional group, and if we are favor with the hydrogen bonding, then it increases the selectivity of your ligand. According to the pharmacokinetic property, we can change the functional group, or we can rearrange the functional group also to reduce the toxicity, to increase the absorption, clearance, metabolism. All this property can be optimized. Also, molecular docking is used for the repurposing of existing drug. After COVID-19, we are continuously thinking of a repurposing of existing drug. And nowadays, it is a most demanding domain of a research where existing molecules are repurposed or repositioned for the newer disease or newer target. Similarly, we can predict off-target effect or toxicities. We can evaluate the natural product and bioactive compound for their binding potentials, which support the discovery of a bioactive molecule from plant, microbes, marine source. Okay, we can explore these other sources of drug. We can use the molecular docking in protein engineering and mutation analysis also. We can check or study the effect of a amino acid mutation on ligand binding, how the ligand changes conformation, and guide and design to improve the enzyme and protein with desired function. We can change the wild type to the mutant protein, and we can study the effect of these changes on a ligand binding properties. In agriculture and environmental application, we can design agrochemicals like herbicides, fungicides, insecticides for a target key protein for development of a newer agrochemicals. Or we can study interactions of pollutant or toxin with biological targets. Okay, so these are the few applications I have cited in this slide. So already we discussed the typical research flow or workflow of how molecular docking has been performed, key benefits, and important or impact across disciplines, and various challenges and consideration. So up to this, if you have any query, you can feel free to ask, or if you have anything, you can write in a chat box. If you have any query, you can ask right now, or okay, there are some questions. Sir, which software is the lightest on my one of my system is low end laptop from 2015. So will it work on my laptop? Okay, okay. Very good question. So most of the people do not have a high configured laptop and PCs. Actually, for molecular docking, a high configured or a workstation is not necessary. If you are using a commercial software like Schrodinger, that might require a specific requirement, like it require a GPU, graphic card. Okay. But if you are using a open source software like Autodoc Vina or Autodoc, that doesn't require a higher configuration. Say for example, i3, i5, i7 and sufficient RAM is or 4 GB, 6 GB or 8 GB RAM is sufficient for running this kind of a docking software. Mostly the open source softwares are lighter one. They can easily run your system. I hope I giving the right answer to the Raghav. One more question is there. So, sir, how do we ensure that the protein selected is a good quality? Yes, good question. So already I explained it. The quality of a protein is depend upon the certain parameter. Say for example, I'll just sharing my screen again. So just look into this site. For quality of a protein, you have to check the first is from which organism it has been derived, whether it is your interested organism. Okay. Then you have to check whether the mutation is there or not. Mostly non-mutation or non-mutated protein are be selected. For your particular study, like you are studying the mutation study, then you have to select the mutated protein. The next, you have to check whether structure is the latest. Okay. It is when it is deposited and when it is published. Okay. The latest one is considered as a ideal one. But again, which is depend upon other factor also. Always latest protein are not always good. We have to check other parameter. Like primary most important parameter is whether it is experimentally, by using which method it is experimentally derived, whether it is from X-ray diffraction, NMR, or EM cryo. Most probably, X-ray diffraction method are suited or chosen for the right or quality protein. Then you have to check the resolution of this protein, how the structure having the resolution. So, sorry, the so the resolution between a from 1.5 to 2 angstrom are considered as a good resolution. So select the protein which having the resolution near about 2. That could be a good quality protein. Then you have to look into this image at your right side, where the blue color and red color is observed. These sliders should be, majority of sliders should be from the bluer color side. Among these, the first and foremost, you have to look a Ramachandran outlier. You might aware about the Ramachandran plot, where the amino acid are distributed between different zones. Okay. In this zone, you have to check the whether the pi and psi, the area is defined in the Ramachandran plot. So the majority of amino acids should not outlier within these areas. So lower the Ramachandran outlier is a good quality protein. So here Ramachandran outlier is 0. Along with that, side chain outlier is also 0. So these two parameter considered as among them, Ramachandran outlier is a good parameter that you can choose for the selection of a quality of a protein. The next, you have to check chains present available, whether you are interested in all the chain or you are interested to A chain, B chain. Then next, you have to check whether any ligand is attached or not. If ligand is not attached, then you have to go for a finding of a binding pocket. If binding pocket or if ligand is attached, then you can take this coordinate of this ligand, and you can choose that coordinate for your binding or for your molecular docking parameter. So these kind of parameter you have to look while you are selecting the protein. I hope I am giving the right answer. Next question by the Sri Charan. Up to what number of range we can set exhaustiveness in our docking score? Okay. Ideally, 8 is a good exhaustiveness score. More the exhaustiveness, give you more confirmations, and it will take more time for a docking. So ideally, 8 is ideal for the accurate binding and accurate conformational, gives you a good binding results. The next question is, what are the software for visualization for a best pose? Okay. Nowadays, Pymol considered to be the one of the most used visualizer. But along with that, there are certain commercial software are also available, or free visualizer software are also available, like Chimera. Chimera is also, or Chimera का दूसरा version है Chimera X, is also one of the good for visualization. Then you can use BioVia Drug Discovery Studio, which is also free for the students and teachers. Then you can use you can use Maestro from Schrodinger, Maestro Visualizer from Schrodinger, which is free for student. Visualizer is free for academic purpose, but docking software is licensed version. So for visualization, you can use any one of this, like Maestro from Schrodinger, Drug Discovery Studio from BioVia, PyMol, then UCSF Chimera or Chimera X. These are the good one for the visualization. I hope I am giving the best answer to Priya. Is Pyrex is equally important as Autodoc Vena? Actually, Pyrex is also follow the same algorithm followed by the Autodoc Vena. There is an add-on in a Pyrex. You can add Autodoc Vena script and everything, and it will run on the same algorithm. So there is no difference between the Pyrex and Autodoc Vena. Do we need a laptop separately as I am attending on a laptop? No. You can do not need a separate laptop. The binding affinity by one more question from Sri Charan, that the binding affinity depend upon the length of the interaction ligand and the protein. Yes, the binding affinity depend upon the length. As I told you that the distance between the two atom or bond length should lie 2 angstrom. The 2 angstrom distance is considered as a good binding interactions. Sir, could you also tell about the software on Mac? Okay, for Mac, most of all the software have a Mac version also. So on their download section, you can check for Windows, for Linux, and for Mac also. So whatever the listed as software that are suitable for Mac also. But some configuration you required, that we will help out you. Sir, why only X-ray structure are selected, not others? Because X-ray structure having the highest resolution. Okay, that's why the X-ray structure has been selected. Can we run complete docking in a one software? So should we use or we should have to use different software? So majority of commercial software are available. All this, whatever the process I explain you, can be run in a entire software. Okay, but that is not in case with the open source software. In open source software, you have to club a different software, and then you have to perform the docking. So if your university or if your college having a facility of this licensed software or commercial software, then there is all can be done in a one go. No need to add compiling of a different software. Is research publication accept this free software? Yes, definitely. We also publish many articles in a reputed journal where we use only open source software, no any paid software we used. Recently, our article published in a molecular diversity with the impact factor of 3.9, where we do a molecular docking, simulation, network pharmacology. Okay, all this done by free software, open source software. No any paid software has been used. Or another thing, you can collaborate with the other people where they have a facility or paid software. So part of your study you can do by collaboratively and incorporate in your publication. What are the minimum parameter to analyze the docking result? Okay, so the parameter which are already explained you that I hope the slide is visible to you. The question is what parameters are used to for a scoring function. So as I told you that the binding score along with the ADMET parameter, pharmacokinetic parameter, all this can be considered for the best docking result or best score. Let me check any other questions. Okay, any more questions? Or you can ask to the organizer for my email address, then I'll answer your question on email also. How docking will be utilized, researcher or doing formulation development? Okay, for formulation and development, there is not only a single docking software can be used. You have to first build the say for example you are looking for a nanoparticle liposome, this kind of formulation. You have to first uh, design the structure of liposome nanoparticle in a software like Material Studio. Then you have to use it for the docking. Sir, will you explain how to draw a structure? uh, the Mr. Jai will explain you how to use a different software, but I'll give you an example of a software like ScamSketch, CamDraw. This kind of software you can use for the drawing of the structure. Please explain docking validation and RMSD interpretation in the docking. Uh, Actually, during the hands-on training of a docking process, I'll answer all the question again. Okay. Once you do a hands-on, then it will gives you idea or understanding how the process has been done, how the scoring has been done, how the RMS value has been calculated, what is the importance of RMSD value. Everything I'll explain during the hands-on. So sorry for the I'm not giving right here answer, but my suggestion is that once you go hands-on, where you can easily understand or easily all your doubt must be cleared. Okay. So let's, if no more question, then we move toward the installation of a various softwares. So I handing over the session to the Mr. Jai so that he can help you out to installing the various software which are necessary for the molecular docking, simulation, and network pharmacology. Over to you, Jai.
Thank you, sir. So, hello everyone. So, for the installation of the softwares which are required for molecular docking, that I am going to present you. My screen is visible.
Yes, visible.
Uh, Okay. So, these are the common softwares which are required for performing the docking and subsequent other perspectives of the uh, workshop, like now for network pharmacology and molecular dynamics. So today I will let you know the softwares which are required for docking. So first and foremost software is Autodoc Vina. I will give you the links of these in the chat box so that you can download the softwares. Okay, so we will go step by step for the downloading some of the software like Autodoc Vina. So we have to go to the website. Okay. So this is the website of Autodoc Vina. Go to downloads, and after redirecting to the site of downloading, we have the options. Some of the participants are Mac users. So based on Mac configuration, you can download the Autodoc configured file from here, and Windows user can use download the Autodoc Vina from here. As I am not able to downloading it, I have already downloaded it, so the software is not being updated in my system. So new user can easily download the software, but in some cases or in some i7 or i10, i9 processors, latest processors, the new update has been there in Autodoc Vina download. So redirecting to the GitHub library of Autodoc Vina, you can download it from here as per your system configuration. Any doubts? Hello. Uh, Anyone is having any kind of problem in downloading any of the software? Please let us know.
Yes, Vibhuti, you have raised your hand. Please share your question.
Hello, am I audible?
Yes, yes.
Sir, I'm not able to download the Autodoc Vina. जो अभी when I clicked on the link, it is showing the same those scribbled words, and I'm not able to download the Autodoc Vina.
Okay. You just follow the process. Just go to 1 minute. You have gone to this website, no? So redirect yourself to the GitHub library repository. Okay. It will redirect to the this website, and scroll it down. And from here, if you are a Mac user, 1 minute.
Windows user, sir.
So you can download it from here. You can download it from Vina 1.2.7.win exe. This one. It will directly download on in your system. So it will be in your downloads. So actually, it will directly run in your system. It will not show any other things. It will directly show the icon, app icon, which I am showing to you. This is the app icon.
I just want to clarify that Vina is a add-on of a Autodoc software. So Autodoc 4, you have to, along with Autodoc Vina, you have to install the Autodoc 4, which is named as a MGL tool. So Mr. Jay, please explain the how to download MGL tool, and
The next,
then you will have to.
Yes, sir. The next chapter is MGL tool. So you need to download the MGL tool via link, or directly clicking the MGL tool download. You can go to this link, and as per the system configuration, you can download it from here. So I'm just downloading it to show you how to perform the downloading process. Based on your net connection, it will take some time to download this tool. Okay. After this tool is downloaded, it will ask you for the control of the system. So click yes. My screen is visible.
Yes, it is visible.
Okay, okay. So when you download the MGL tool, it will ask you to the next. I accept. Then yes. Then it will save your file in the location in C drive, program files. Next, and so complete it. After. So I have already downloaded it, so I am cancelling it now as it will replace it. Okay. So after downloading this, you may see the other tools, 2, 3 softwares along with the Autodoc. Okay. This is the other software which is required. After that, Open Babel software. This is the software. Means I want to tell you that Autodoc Vina is the visualizer or the Python based interpretation of the all the docking scores or the all the Autodoc based docking parameters. So MGL tool are the main tools which are used for, as sir has explained, that some force field are there which are used for the docking scoring. So MGL tools has all these parameters. So with the help of both of these softwares, we can use it for molecular docking purpose. Other software is OpenBabel. This
Okay, just just
OpenBabel's,
just just wait,
yes, yes, sir.
Jay. I'm just explaining.
Yes,
So what I am trying to say that MGL tool is a it will help you to install the Autodoc 4. Okay, while Autodoc Vina is a script based or Python based tool which will help you for the virtual screening. Okay, by using MGL tool, can you see the screen, share the screen for the Autodoc icon? Jai, please redirect to the Autodoc icon
oh yeah,
on your desktop. Yeah, just click on it and open it. So once you click on the this Autodoc tool, it will open the window. Okay,
it
I'm
will run a
giving you a
Python script.
it will. I will explain you difference between MGL tool or Autodoc and Autodoc Vina. So this is the GUI of a Autodoc. Okay. So here the limitation of this Autodoc for GUI of this Autodoc is you can uh, do a molecular docking of a single molecule only. Okay. If you have a n number of molecule, you have to perform each and every step every time. Okay. So for that, the Script Institute had developed this Autodoc Vina Python script, which will help you to a virtual screening of a n number of molecule at the same space or same time. So for that, that's why we are using Autodoc Vina along with the Autodoc 4 algorithm. So that is the main difference between Autodoc 4 or MGL tool and Autodoc Vina. For virtual screening, you require Autodoc Vina only. If you're having n number of molecules you want to screen, then you have to use a Autodoc Vina. For a single molecular docking, less number of molecule, you can use a Autodoc 4. Okay, I think I clear your doubt. IgM doc also can be used.
Yes, they can be
It
used.
is also a similar kind of a Python script based docking. Go ahead, Jay.
So after this, the others another software is OpenBabel. So OpenBabel is the software which is used for the conversion of file format to different file formats supported by the Autodoc Vina or any other formats. So this will help you to easily convert the files to its format required by the software. So specifically for Autodoc Vina, the PDPQT file format is required. So we can convert it easily with the help of OpenBabel software. So I have provided you the GitHub library link. So go to the link, and after that, you have to download it, source zip file, and extract it.
Okay, meanwhile installation, I'll just answer the few questions which is asked by the Sivraj. Say, my laptop is closed immediately. So
RAM.
due to maybe a RAM issue,
Yeah.
what is the configuration of your laptop? I think Sivraj is, it might disconnected.
Hmm. Actually, this will take some time. I will let you know, extract it, and after extracting it, run the exe file. Hello.
Yes, you can continue.
Yeah. Or else you can read me software you can use, read me tool, so that you can get a better understanding that how to install it. So actually, I have the older version, so it is not allowing me to download this zip file. So what you can do, you will get uh, a software to be installed in this logo or in this format. So this is the OpenBabel software icon, and after successful installation, it will show a UI interface like this. So where you have to uh, have various input options, and convert it into a various output options. So mainly the structures which are available in PubChem are we can easily extract it out in smiley format or SME format. So these are available, which can be easily converted into a Autodoc representative form, which is PDPQT format. Is can be converted easily by with the help of this software. So in case the software cannot be installed, the OpenBabel can also be converted online based also. So with the help of online converter, we can also convert the file format also. So this is the online interface to convert the file format. So for this, we need to download, we can provide the smiley as there is the example, and I want to convert it into a PDPQT format. Okay. So I can convert it into a PDBQT format. So at the time of docking session, I will let you know how to save this file. For now, I will tell you that just copy with the help of Control C or right clicking it by copy, and here go directly to the system and open Notepad. After opening the Notepad, just paste your this data of PDBQT and then file, save as. Okay, it will let you know the folder location. Let the UI give you the folder location. After that, please select all files here and rename it as compound. Your target compound name dot pdbqt. And save the file. It will directly save the file in pdbqt format, as you can see here. As the given compound is directly converted into pdbqt format. So these are the well-known compounds which are available at the PubChem database. Okay, so we can in the case of OpenBabel, we have the both the option, software option for the offline purposes, and for online purposes we have the online UI interface also. Moving towards the next software, so this is the Pymol software used for the visualization. So this is the small molecule which is example which is given in this
Jay,
software.
hold,
Yes, sir.
just hold.
Yes, sir.
Few questions are there that let me answer the questions.
Sure,
The
sir.
many people are facing issue while installing the Autodoc Vina. Your laptop is getting shut down immediately. So I'm just want to clear that you not need to install it. Autodoc Vina not need to install. Just keep the file as it is ready.
Yeah, you do not need to download the Autodoc Vina file. I will
Install,
provide
not a
you.
download.
Install the file. Make sure that you have the exe file in the folder. Okay, so I will provide you the folder in which the required material are there. So directly from the folder, you can run the script. It will not be a problem. Okay, so after OpenBabel, the next software is Pymol. Pymol software is the software which is license based and free also. So you can download it from this website based on your system configuration, and it will be downloaded in your system. And for license academic version, it is free. So if you have any kind of academic email ID or academic material, so you can use it for downloading the academic version.
Let me just clear your doubt here. The actually Pymol is a previously a open source software, but now it is acquired by the Schrodinger. So previously it was a freed one or open source. After acquiring by the Schrodinger, it will become a paid. But for academic and student, it is freely available by providing your authentic institutional email ID. You have to register yourself in a website. Once you registered, you will get the license for 1 year. After 1 year, you have to again renew it for free of cost. If you have a official email address, then you can easily download. Also, various open source available Pymol version is also available. You search out on Google, you will find the open source version also. Few questions are there. Is Autodoc tool 1.5.7 icon the one that will appear after the download Autodoc Vina?
Yes,
Again,
yes.
again, I'm clarifying it. clarifying it. The Autodoc Vina will not gives you any icon, but Autodoc tool will give you a few icons on your desktop. Okay, just keep in mind.
In some
Repetition
cases,
of, sorry, for repetition of
yes,
open babel during the you molecular docking process, we will repeat all the step by using how to use open babel. So no need to worry about it.
and if you want to down problem in downloading, so you can also use the OpenBabel in online mode also. So it will be not an issue that OpenBabel is there in your PC or not. Okay, and another thing, the Autodoc downloadation only requires the exe file only, not the other files also. Okay. Moving towards the next software, which is Pymol. As sir has explained, that Pymol now has become the paid but free for institutional use. So you can download it from the link. Another software is Discovery Studio. Actually, Pymol and Discovery Studio, these are the two softwares which are used for uh, visualization. So another software is Pymol. So another software is Discovery Studio. This Discovery Studio is not supported by the Mac users, so they can use the Pymol. So for when you go to this website, you first need to register yourself with the help of your email ID, and after an hour or after a certain period, they will answer you within 24 hours. They will answer you via mail and give you the download link. Then download it with the help of the link, and it will directly the software uh, icon will be appeared on your desktop in this format, Discovery Studio. And the UI interface of the Discovery Studio is like this. Based on your system and based on your system's RAM, it will take some time to open the software. So this kind of interface will be visualized with the help of Discovery Studio. So one another thing which is required for molecular docking is Perl script, which is Perl IED. So you simply go to Google and just search Perl IED. It will show the blue color butterfly icon. Go to the link and click download. Click on download. And based on your system, download the script. So this is for Windows. I am using Windows. The Linux users can use the Linux system also. Okay. So here download this one. After downloading it, just confirm that your download has been completed or not. By using command prompt, you can easily get know that the this software is installed or not. So simply go to the Windows icon and type
Jai,
cmd.
answer the question first.
Okay, I will let you know. No, this is the system. I will provide you the script in written format. Actually, I am not familiar with the Mac users. So actually, Ubuntu system is there, but no Mac version. Yeah, Mac version is there. So you can download it from the GitHub repository provided over here. If you have any doubt regarding this, please, you can take directly the script. In your command prompt, you directly run this script, which will directly install your software. And if you are facing any type of problem, so you can readme is there. Yeah, you can use this script to download in your Mac system. Actually, Mac downloadation can be done with the help of coding. So you can download with the help of this readme document. Okay. So this is the another Perl script is required for performing the docking of multiple ligands with one protein molecules. So as sir has told you that with the help of Autodoc Vina or Autodoc 4, we can dock one molecule at a time with a one protein molecule. So in case with the help of this script, which is developed here, Perl script, which can be used for docking of multiple ligand molecules with the single protein molecule. So it will reduce the time and give you the result same as that of single molecule docking at a time. So these are the main softwares which are required for conversion and the running of the docking. So if you have any doubts in downloading the softwares, please feel free to ask.
So I request all the participants to download and install this software and be ready your laptop for the hands-on training so that you can match up with our speed. Or you can get the better idea how to perform the molecular docking by using this software. Yeah, we will provide you the protocol for how to perform the molecular docking. Our team will help you in providing this protocol.
It will be demonstrated and performed at the time of docking scheduled.
So Vibhuti, which system you are using? Windows or Mac? Windows. So you are facing issue like installation of error occurred during the installation of assembly compartment.
Okay, that is the. Can you please share the screenshot? Uh, Most probably it will be the problem related to Java. So some cases it may not be installed in Windows because the Java version are different. So you can download the older version as you have now you have downloaded the 2025 version, so you can install the 2021 version for your system.
You can share your screen and you can also
Yeah.
share your problem. Even now we are also using the older version. We are, as you write, you might see the in a Jai's laptop, we are using the Discovery Studio 21 version. We are not
Yeah.
using the latest one,
Sorry, actually
because
my
sometimes the latest software may not work. So whatever the
there are
older version are optimized one. So you can
yeah
use older optimized version also. That also valid.
uh, actually there are some bugs in 2025. New molecules which are not there in PubChem cannot be visualized with the help of this tool. So it's better that you should use the older version which are compatible with all other softwares also. uh, Bhagyashree, Pulse script. Yeah, Pulse script I will provide you at the time of docking. I will provide you the uh, folder which will be directly used for all the docking process. If you want to do a docking of for a one protein molecule, so you need to copy the whole folder over the file directory and use it for single protein molecule. For another protein molecule, you need to copy the same folder and perform the another dock. Okay, so
Okay, I think Mr. Jai have completed the all the software related to the molecular docking. Jai, anything left?
no, related to Autodoc Vina and Autodoc docking is completed. And related to Open Babel, I will let you know at the time of docking hands-on training.
Now I request Mr. Bhunesh. He will guide you to install the network pharmacology softwares. Please, Mr. Bhunesh, over to you. Hello, Dr. Sumit.
Yes, sir.
Can you ask to the Mr. Bhunesh for
Sure, sir.
yeah. Just wait a minute. He will might be joining the session. Meanwhile, if you have any query regarding the installation of your software, you can just ask. So meanwhile, that Mr. Bhunesh can join. Yeah, the one question by Vibhuti that, sir, only one option is seen for the download for the Discover Studio, no other version. Okay, let me check and I'll share the link.
Bhuvnesh sir.
Hello.
Yes, sir. Please start.
Hello. Good evening. Good evening, students. All students, please download Cytoscape software, which is used in network pharmacology. If you have any trouble, so please put your comment in comment box. Completed or not? Then further, two plugin install in your computer and laptop. First one is Gene Mania plugin. Gene Mania plugin. And second one is Cyto NCA plugin. You can directly install through Cytoscape software. Go in Cytoscape software and download Cytoscape Gene Mania plugin and Cyto NCA plugin. Gene Mania plugin will use for network construction and Cyto NCA plugin will
I think he might be disconnected. So let me just share my screen. You can go to the software, go to the website and download this version according to your requirement, whether you are using Windows or web. There is a web version also available, but I encourage to, instead of using web version, use a download version. You can use according to your system. Download the Windows version and it will download it here. I'm just interrupting it because I've already installed the cytoscape in my system. So
Oh,
I'm just
thank you, sir.
opening it. Installation is very easy. Just next step is for the installation. You can use any version of the software. Okay. Once it will load all the plugins and everything, it will little bit of take time to load the software. It is totally Java based software. So this one is an interface of a cytoscape. Okay. So from app, you can use a various plugin. Okay. By default, you have a certain plugins. Okay. If not, you can directly install from the file which is you have downloaded from the website, or you can directly uh, go to the website. If you are downloaded already plugin, you can install from whatever the desired location. I'll let you know how to download the plugin. So write the Cytoscape plugin. It will open the website where all plugins are available. So as the Bhuvanesh sir said, you have to download Gmania. Just a minute. So this plugin is you can download from this website. Already I have installed it. So let me just explain you how to download it.
Sorry to interrupt you, sir.
Yeah.
Directly search to Google Gene Mania plugin and you can
Okay.
install easily.
Okay. Yes. You can guide, no issue. Bhunesh sir, I'll explain everything online. No issue.
Yeah, thank you, sir. Okay, next, Cyto NCA. Similarly, please download.
So using this interface, you can use it, or you can download from the app also. Just yes, yes, Bhunesh. Which plugin?
Yes, Cyto NCA. Similarly, download Cyto NCA software.
Cyto
Cyto NCA.
NC.
NCA.
Yeah.
Yeah, this one, please download.
You can download from here.
Yes.
The plugin will download in your download folder. So now you want to install this plugin, go to the software, go to the app and install app from the file. Relocate your download. and find the plugin. Sorry, just let me check then.
Yeah, now completed, sir. Is
Okay. Okay.
yeah. You can check all students. You can check easily in your Cytoscape software, and now you can perform your practical. So tomorrow and after the we will perform by the Cytoscape software network pharmacology. If you have any trouble, so please comment in comment box.
If any query, please feel free to ask the or contact us for any troubleshooting. Yeah, team will share the today's and yesterday's recording to you. Yes, I am checking it. Once I'll get the information, I'll share in a group. Vibhuti, I'll share information in a group.
Okay, Sumit sir.
Yes, sir.
Now completed all or not?
Yeah. So, good evening, everyone. As we come to the end of today's session, I would like to sincerely thank all our speakers and participants for making day 2 a great learning experience. Our heartfelt thank to Dr. Ghansham for delivering an excellent session on the fundamentals of molecular docking. We also sincerely thanks Mr. Jamukesh and Mr. Bhunesh for guiding everyone through the software installation process with patience and clarity. We also thanks all the participants for your active participation and questions. We hope today's session has helped you prepare for the upcoming hands-on practical session. On behalf of Thyme Phytobiomed Private Limited and Edgar Foundation, thank you once again for joining us today. We look forward to meeting you again tomorrow. Thank you, everyone. Have a wonderful evening. Good night and Jai Hind.
Thank you all.
Thank you all.
Thank you all.